Welcome to the Dr. UUT channel. Today's topic is cellular transport. This video is in English and available in Urdu. Let's proceed in English. Topic: cellular transport. First, let us understand what the term means. Cellular is obtained from the cell. Transport means to do export and import. Means that we have a cell, something moves in or out from the cell is known as cellular transport in short. We have cellular transports of two types: endocytosis, exocytosis. First, let me clear the terms again. Endo and exo and cytos. Endo means inside. Exo means outside. Cytos is obtained from the word cell. So cytos means cell. If something is moving inside the cell, that is called endocytosis. While if something is moving outside from the cell, is called exocytosis. Exo means outside, cytos from the cell. Exocytosis, something is moving outside from of a cell. Well, endocytosis and exocytosis clear. Now endocytosis is further divided into three: pinocytosis, phagocytosis, and receptor mediated endocytosis. These three are put in the endocytosis for the reason that because in all of them the process that takes place is the things are moving from outside to inside. Things are moving into the cell. Okay, that's why these three are put in the endocytosis. Now, let us clear each. Pinocytosis. You know, cellular drinking and eating. What is cellular drinking and eating? As humans, we eat and drink. The same is the case with our cells. They eat and they drink. Well, if something is being eaten, we'll put that in the phagocytosis. Well, if something is being drunk, that is called pinocytosis. How? If a liquid is brought inside the cell, or if a liquid moves into the cell, that is called pinocytosis. Pinocytosis is if a liquid enters the cell, that is called pinocytosis. Means cellular drinking is called pinocytosis. So what are the cells involved? Very simple. Our kidneys, our liver cells, they are very important, prominently involved in the pinocytosis. And epithelial cells, they undergo the process of pinocytosis, means they drink. Well, what is phagocytosis? Now, it is called cellular eating. How? You know, we eat, we drink liquids and we eat solid. So, cell also eat and drink. Eating is phago. In this, what will happen? Something solid is brought into the cell, or solid way if enters the cell, or if eaten by the cell, taken inside by the cell, that is called phagocytosis. What are the cells involved? The very simple example is of macrophages, like neutrophils, basophils, etc. These are called, means macrophages in short. They are responsible for the cellular eating. They eat. How they eat and how they drink? So simple. First of all, they will encapsulate, means they will cover in the capsule if it is liquid or a solid. After that, they will bring that to the cell membrane. Then that capsule will fuse with the cell membrane. After fusion, whatever is present inside will come from the cell, that vesicle, to the cell. And the same is the case with the solid. Then what will happen? Then cellular metabolism will happen. Whatever needed is will be used by the cell, and the excess will be secreted out, means they will be excreted. That is called as exocytosis. So there, whatever the remnants are present thereby, which are not going to be needed by the cell, they will be removed out, or they will be moved out by the cell. That process is called exocytosis, means outside the cell. Well, I will make this clear again with another example. Another one is receptor mediated. Now, what is this kind of transport? Very simple. What happens in the receptor mediated? First, concentrate receptor mediated endocytosis. This is a kind of endocytosis that is done by the receptor. We have a cell. Here is a receptor. So if something comes, attached with this receptor, then moves inside, that is called receptor mediated. Let me give you example. Well, we have cholesterol. Cholesterol is further divided into LDL and HDL. LDL, I'm talking about that, and I will not go and not explain further what is LDL, HDL further. Now we will first let me clear about this receptor mediated endocytosis. Well, LDL, not HDL, LDL. LDL will come, and this LDL further will be enclosed by the lipid bilayer, phospholipid, and a protein is attached further with that LDL. Now LDL is now enclosed by the lipid bilayer, and a protein is further attached with that LDL now, encapsulated LDL, you can say. Now this protein is responsible to attach with this receptor that is specific for the cholesterol, okay, LDL. Now this receptor and this protein, they both will combine. After combination, this receptor that is attached with the cell, this will be activated. Now this activated receptor will send the signal inside the cell. There are further two types of proteins, adaptin and clathrin. First of all, adaptin will come and will attach here. Then the clathrin will come and will attach here. Adaptin will encode and clathrin will do what? It will invaginate. It will bring, it will, you can say, pull down inside the cell. How like? Now, this is the receptor present at the membrane, on the membrane. Now, this is the LDL covered with the protein attached by means of adaptin and clathrin. Now, this receptor will be invaginated along with the membrane. Now, membrane and receptor, they will be invaginated, will be pinched inside. You can say, will be pulled inside. Then what will happen? This will be like that now. This like this. This is called invagination. When the membrane moves inside, it's called invagination. This is done by these clathrin and adaptin. Adaptin will attach and clathrin will further invaginate the membrane or will pull the membrane inside. Like that, then these the marginal membranes, they will rejoin, and this one, the one that had the receptor and the LDL, that will come here. Now this is LDL and receptor. I'm mentioning the LDL by L and receptor by the symbol of receptor. Now this will be further targeted by the intracellular enzymes, etc. Then again the receptor will be free and the cholesterol will be free. This cholesterol will be again used, and the same is the case with the receptor, will move upward, will be upregulated, and will come on the surface of the cell to bring inside more cholesterols, and the cholesterol that is inside the cell will be further used for different purposes. You can say for a purpose of formation of the membrane when there is a need. So this is the mechanism by which something is brought out from outside to inside by means of the receptor. This mechanism is known as receptor mediated endocytosis. Got? Okay. Let's move towards the exocytosis. What is called exocytosis? Very simple. I will give you an example of neuronal cells. Here is one neuronal cell and here is another one. They communicate with one another by means of neurotransmitter that is enclosed in the vesicle. Here, I will mention these by the dots. So what happens? Now the vascular substances the substances that are enclosed by the vesicle will come here and will be moved out. That is called exocytosis. How will it happen? By the two ways. First of all, action potential is generated. Means a signal is given. I'm making you people understand in short. Well, these are very complex processes. For the sake of exocytosis, I'm just making it short. Action potential will be generated by which the signal will be given to the vesicle to move towards the end of the neuronal membrane to fuse there. Now this, when it reaches here to the membrane, in order to fuse with the membrane, this process of fusion is done by the calcium and by means of two proteins. By means of proteins, two types of proteins and calcium. Like how? Very simple. The calcium will help these two proteins. The proteins that is present on the neuronal ending at the end of the membrane and on the vesicle, the proteins are present. Now these both proteins will combine by the help of the calcium, because calcium has got two positive charge. So this will be, they will be brought together, will be fused. Now when these both proteins combine, after that what will happen? It will be fused. After the fusion, its responsibility is to release the neurotransmitter. That is released and will then come. Here again the receptor is available on the postsynaptic neuron, means on the another neuron. Then that neuronal receptor will sense the signal that is being given by this receptor. Then again the mechanism will be with this receptor. So this is called exocytosis. Means something is moved out. That is called exocytosis. How is it done? By the action potential generation by the means of calcium and by means of two proteins. That is called exocytosis, and the name of the proteins are synaptobravin and syntaxin. The protein available at the vesicle is synaptobravin and available at the membrane. On the membrane is syntaxin. These two will be combined by the means of the calcium, and after that, when the calcium will bring those together, these two proteins, and will combine them after the fusion combination, then there will be a kind of fusion with the membrane, due to which there will be the release of the neurotransmitter. This is called exocytosis. Something removed or moved out from the cell. That's it. Very simple. Endocytosis and exocytosis. Hope so. You got it.